So now I'm going to tell you about how to get barley seedlings started in our greenhouse. First, I want to start off with mentioning that the module that we will be using this spring is one that was created at the Dr. Roger Wise lab at Iowa State University, and it was done through the auspices of a National Science Foundation grant. One of the reasons we are able to do this module here at Ames High School is my colleague Mr. Walter and I have spent many years participating in this National Science Foundation initiative to bring modern day biotechnology aspects to today's classroom. So, many thanks to the National Science Foundation and the Dr. Wise Lab, and on to the details of how to get started. So we're assuming that you've got some properly prepared potting mix, a pot of approximately 5 to 6 inch diameter. Take that pot, scoop it right into the potting mix, and we're going to fill that pot right up with the soil mixture. And we're going to get this so that the soil is pretty close to up to the top edge here. Now, most of our pots have what's called a lip right here, and that's where we want our soil surface to be. So if you fill a little bit above there, you can gently, and I do mean gently, tap that soil in there. We're not trying to squeeze all of the air out of the soil. We don't want to compress the soil like that, but we want our soil to be up near the top of the pot so that the pot doesn't shade out the very plants that we're trying to get started. Planting depth will vary depending on what your crop is. We will be using barley because of the genetics involved. We have seed that we know will have certain traits that you will be easily able to see in about 2 months as these grow out. These traits, of course, are coded for by a gene, and what we're going to try to do is show that connection between what we can find with modern biotechnology tools and the genetics that you can actually see with your eyes and make that connection. The planting depth for barley is about 1 inch or about 2.5 centimeters. I like to measure with my finger, and from the tip of my index finger to the first set of wrinkles here, or that first knuckle, is about 1 inch. So I just take my pot, take my finger, and put it in about 1 inch deep. We're going to plant 2 seedlings with the idea that our germination is likely to be high. We will probably eventually narrow down to 1. Here is my barley seed, and we're just going to place that in one of the holes, cover it up, and now we're ready to prepare our label. We have these popsicle sticks as our labels. I'm a big fan of redundancy, in other words, repeating labeling. So you're going to need 2 major pieces of information on the popsicle stick. One will be the number of your table group in my class. So I'll be filling this out for one of my first classes of the day, period 2, and I'm simply going to label with a 21 for them for table 1 in that class, period 2, table 1. We've been through this instruction before, so you should know how to apply this label. I'm going to suggest that we label on one side with Sharpie, but since these will be getting watered, pencil may be a more reliable method. So on the other side of the stick, I am going to label in pencil that 21 for table 2-1. The other thing you'll need to know is which variety of barley you have, your teacher will tell you. In this case, I'm working with variety number 2, and therefore, both in pencil, I would write number 2, and in Sharpie, number 2, like so. And now, near the edge of the pot, we will just place that in the pot. For redundancy, I've got some little pieces of tape I've provided each group with. Again, we'll use a Sharpie 21, number 2, and below that in pencil 21, number 2. And we will place that label right about there on the pot. And then follow your teacher's directions on where to place these in the greenhouse. We will be keeping them lit for about 16 hours a day, so they will be under our fluorescent grow lamps. And thank you so much for listening to our video about how to start this